Well, I hit it, rejected it. You're doing it. What does that mean?
Hit reject. It's like somebody's trying to call in, I guess. Was this supposed to be, this is just recorded, right? It's not like a conference call?
Right.
Just hit reject on that.
Yeah. It came up right as soon as the radio started, so it was just old. Yeah, it's not about it. Anyways, we can go to... I mean, it's recording, so, and you won't see that there.
I wanted to get one. I'm surprised we had just like
Oh, okay. Yeah, I mean,
Yeah, set computer is a button. Put yourself up there. Does that? Yeah, oh no, that's good. Is that? Oh, it's that's amazing. And all the talks are there.
they're in different.
I don't know what they're open. It's like the video.
Because we were just everything was set up, it's already recording. So now I can go to lighting and adjust the stuff. And it's gonna that's kind of
I'm not gonna like put there.
So welcome to our first ever Disasterpiece Theater. The last couple of years we've started off the seminar series featuring faculty speed talks. This year we decided to change it up and try something else. True story, when I made the logo for this, it was featured in the flyers that got sent out. I spelled it Disasterpiece Theater. It's kind of a funny joke, but yeah, you decide. So that said, anyway, I'm the co-organizer of the seminar series and do this for several years along with Laura Gigliotti and David Slab. She had this great idea this year that instead that a lot of faculty might have like stories that they'd like to tell or maybe research that they're embarrassed to share, so why not ask them to do just that? This is partly inspired by, if any of you guys are on Twitter, you may have heard about the fieldwork fail movement. So it's become quite in vogue to share our failures on social media particularly. They make for good stories, especially with cool visualizations. Like, for instance, Alistair Dove, who skillfully applied a $1,000 satellite tag to a manta ray, the exact same manta ray, they get tagged the previous day. Fieldwork fail. So these are really funny and make for good stories. It kind of leaves you wondering, well, what happens if maybe all your tags fall off? Like, what do those people just quit science? Do they lose their jobs? So it leaves us kind of wanting to know the rest of the story. So, and on that note too, some of the greatest inventions in history, like cookies and penicillin, have happened by accident. So I don't know if you know the background of penicillin. Sir Alexander Fleming, I just looked this up like 3 minutes ago. Do I remember it? He was studying Staphylococcus bacteria, or Streptococcus. And he came into his lab one day and found that his bacterial colonies had all died. That's because there was a fungus growing on them, which is now penicillin. And then perhaps an even greater discovery was in chocolate chip cookies. The starter, I guess, of the Toll House franchise, she had run out of cocoa baking powder for a cookie batch, and so she wanted to see if she could get a chopped up chocolate bar to melt in cookies. Well, the bad news is it didn't melt. But the good news is we have chocolate chip cookies now. So that said, so even though a lot of great discoveries can happen by accident, sometimes we're left in a position too where maybe the project that we're working on, we poured a ton of money into, is testing out a hypothesis. And maybe over time, with some interaction, we get a certain response that we predict. Then sometimes reality, we can get just the opposite or no results. But maybe that could turn into something greater, like we actually discover some cool mechanism like that. So anyway, today's purpose is to show you the rest of the story of what happens maybe after what you perceive as failure and actually not being failure, and you can still go on to do really cool things like teach at Clemson. So, really amazing things. Okay. So to start us off with some stories is Dr. Pat Jonas from the co-op unit.
Thanks, Abby.
No presentation.
I was going to make slides, but I afraid they just end up off. So, well, thanks for doing this, Abby. Thanks for organizing. And I know you've got some other faculty lined up as well, so I'll try and keep this relatively brief. Abby kind of told me what she wanted to do, and it was funny. I was talking to Troy and Beth, and we're like, we have everybody who's been doing this has got stories about stupid things they've done, things that haven't worked, near disasters or bodily harm or actual disasters. And then there's also the subcategory of things that happen to your technicians and graduate students that you didn't hear about until after the project was over. Like, you could do one of those also. Like, well, I didn't know for 2 years that had happened. And so we have those as well. So I'm going to just go over 3 things that I encountered at 3 different stages in my career, and all of which actually turned out well. And I think for grad students, hopefully there's some good, just some good motivation in there that sometimes the problems that you face, you can actually turn into to good things. So during my master's career, I was a grad graduate student at the University of Florida, and I was tasked with studying the ecology of fox squirrels in the Big Cypress National Preserve. It's a really unique population. They're in a really unique system. Hadn't really been studied before. Fox squirrels have been studied elsewhere, but not that subspecies down there. So it was a really kind of high-risk project, and we didn't know much about them. So I went down to the Big Cypress in January of that year with a plan to be down there for about 6 months. And after 6 months, I saw 6 squirrels. And so when I came back to Gainesville to have a committee meeting and reported that I had seen 6 squirrels in 6 months, my committee, in an understated way, just said, well, that's not going to work. Right? I did a thesis out of 6 squirrels. Like, what are you going to do? And I put some homework in before I went back to the meeting. And I said, well, I have this other idea. I said, it seems like that, there's this thesis that I found for this other grad student somewhere else, and they noticed that there were a bunch of fox squirrels on golf courses in this other state. And, and then they just sort of reported on that. There wasn't a lot of data, and it was kind of interesting. I said, I was thinking maybe I could go to the southwest coast of Florida. There's a bunch of golf courses there, and just see what I could see. And that was it. That was the extent of my idea. Like, I could just go there and see what I could figure out. And they're like, okay. So I did that, and I ended up spending another year in the field. So I had one field season, went 18 months. But I went over to the golf course 1 or 2 days a week. I spent the other 5 days a week in the Big Cypress trying to follow through on that contract with the National Park Service to help them figure out anything we could about fox squirrels there. But by going to the golf courses, I was able to look at diet and behavior and learn a lot about those squirrels in a unique setting, but one that was really becoming common in Southwest Florida and elsewhere. That worked out really well as a core of my thesis. I got it published in the Journal of Wildlife Management, and I won't say how long ago that was, but it was quite a while ago. And that paper still gets cited. Every couple of years it still gets cited. So that was something that could have gone terribly wrong, but I had a committee that believed in me and gave me some support and gave me some space. I had sponsors in the Park Service who said, yeah, if you want to go over there a couple days a week, that's fine. Just keep getting lost out in the swamp and finding squirrels when you can. And we were able to piece that together. And eventually, because of the work we did on the golf course, we also figured out things about squirrels that we were able to translate back to the Big Cypress and do some work there in a translocation project that actually allowed us to begin to understand better what goes on with squirrels in the Big Cypress. The other part to that that's really interesting is that led to another PhD student at the University of Florida following up on that project, which I was really proud about. And it led to my second graduate student here, and Susan Lowe was on that student's committee, because I carried around those ideas for 20 years and was able to get money to do some work here in South Carolina on fox squirrels on golf courses. So that was something that had a chance to be a real disaster, and God knows I lost a lot of sleep trying to figure out where squirrels were in the Big Cypress. I literally used to have dreams about, like, walking into some clearing in a prairie, and there literally would be, like, dozens of squirrels. And I'd be like, I found them! And then I'd wake up and be like, oh, man. That's an actual thing that happened. So that was that. So during my PhD, I had another high-risk project. I was working on marbled murrelets, a small seabird in the Pacific Northwest. And had gotten money to put radio tags on them so that we could catch them at sea, and I could do a whole hour just on capture events at sea in a small zodiac in the Pacific Ocean, how I was grateful to survive half the time. But the idea was we would radio tag them at sea and then follow them inland and find their nests and really begin to understand their nest site selection. So that didn't happen. We tagged 15 birds in 2 field seasons, which was not very many. None of them went inland ever. I did once. I was flying once, and I had to signal inland, but it was an elk. It was just on the same frequency. So, but I didn't know that until I got back. So I thought I had discovered a nest, and I didn't get anything. So anyway, I'd spent all this money on that. It was supposed to be a big part of my dissertation. And same thing, I had to figure out kind of what to do. So I realized, like, oh, okay, these are just standard VHF tags. When these birds dive under the water, we lose a signal. I can use that loss of signal to measure dive times. Again, I didn't put a whole lot of thought into it. I'm like, I can get dive times. If I can get dive times, I can get something out of that. Well, as time went on, I kind of thought about it more. I did some more reading. And it turns out there was this whole series of people who were out there studying dive times in birds and looking at dive times and pause times and diving physiology just based on knowing the rhythms of these dives. Well, I turned this little idea of just getting dive times and pause times into a chapter in my dissertation. Included this whole physiology component that I had never thought about before until I started drilling down and really looking into the literature. Got that chapter published in the Canadian Journal of Zoology. It's the most cited chapter out of my dissertation, still gets cited regularly. I still get asked to do paper reviews on diving behavior in birds and diving optimization. And that paper, when I presented it at an AOU meeting, was the reason I got offered the postdoc that I ended up taking. Because the guy who saw it said, hey, you're really interested in physiological ecology, I've got a postdoc for you. And I thought, I am? I didn't know I was. I didn't know it was a thing I could do. So, again, that was another really serendipitous event where a little bit of hard work and, again, some flexibility on the part of my committee and believing in me that I had an idea that would pay off worked out really well. And it's become a core of some of the things that I'm still interested in science that I'm teaching in the physiological ecology class now. So as I said, that led to my postdoc, and this will be my last story, and I'll move on. For my postdoc, I was studying reproductive energetics in seabirds in the Gulf of Alaska following the Exxon Valdez oil spill. We were doing doubly-labeled water work, actually measuring energy expenditure in kittiwakes. And we had this idea after about a year that if we could also, at the same time we were measuring their energy expenditure, if we could put radio tags on them, we could get some people in a Boston whaler to follow them around Prince William Sound and see what they were doing. They have their own stories of survival, which are pretty good. But we could combine those two things, and we could not only know how much energy they were spending, but we could know how much they were spending while they were doing very specific activities. By doing that, we could model the energy expenditure for what we were calling activity-specific metabolic rates, which very few people have been able to do. So we had a plan for that. It was really well thought out. We got out there in the field. We began to execute it. And after the first, let's say, third of the animals that we were going to work on, after we had those out, because we were following them all the time, we found out that their behavior immediately after we dosed them with doubly labeled water was not normal. Like they were doing stuff that was a little bit abnormal because of the holding period. After you dose them with this isotope, you got to kind of hold them for about an hour. And we thought holding them and screwing them up. They're not behaving normally. And so our data, where we're trying to measure activity-specific metabolic rates, their activities aren't normal activities. We feel comfortable about what we're measuring them doing, but they're not doing the normal things we want them to do. So what do? Well, my postdoc supervisor wasn't there, and this was sort of his project. He had gotten the money. I was trying to run it. So in the field, my colleague and I, this guy Rob Surian, who's still a very good friend of mine, we sat down in Shute Bay, Alaska with a bottle of scotch, and over a couple of days, without field computers, with pen and paper, we figured out how we could change the study design, change the handling procedures so the birds would act more normally as soon as we let them go. But then we had two data sets that weren't collected the same way. How could we bring those together in a way that we could still analyze them and publish those data? And again, with pencil and paper and a little bit of whiskey, we got that figured out. Figured out how we could do what we needed to do, how we needed to bring those data sets together and merge them. We did that. Again, got that paper published, and it still gets regularly cited, and it's a really important piece of work. So in all three of those events, I don't want to make it sound like, oh, I was brilliant, I came up with a great idea. That's not what happened. I was around good people, I had good inspiration, I had people who believed in me and trusted me to try and do something a little bit different. And the key was we persisted and we didn't give up. I think all of us in our research, something goes wrong all the time. And a lot of the time you're out in the field by yourself, and you need to figure out how to fix that. It might be something like a broken down engine, or it might be something bigger like my data aren't going to get collected the way I think. And so a lot of it comes down to how quickly can you work on the fly, what kind of support team do you have in place, and can you make it work? And I think you're going to hear a lot of other stories because you can make it work. You need to believe in yourself, you need to believe in the team around you, you need to stick with it. And that's a big part of what we do. You do that day after day, week after week, year after year, and the next thing you know, you got a career and you're standing up here in front of a bunch of people telling them how lucky you are and that you're able to pull stuff off. So that's my story. Thank you for listening, and I'll let you introduce whoever is next.
Hi everyone, my name is Christy Sampson and I am a postdoc in Sherry Rodriguez's lab and a Smithsonian Fellow where I've been working for about 9 years, and my first fail was naming it the same as Abby's presentation so that it got written over. So just going to quickly put this in, but if there's a light. When Abby first asked me to do this, the first thing that I was thinking about was everybody has medical fails in the field, right? So I was going to talk about either something like when my coworker fractured her femur when we were deep in the jungle, or getting dengue fever, or the only time that my entire field crew was in Sri Lanka together, I managed to get quarantined for about a week and missed all of the meetings everyone was having. And then second, those unexpected skills you're going to need when you go into the field. I'm horrible at climbing, and apparently I did not realize it was going to be a really big part of my project. So, at the end of the day, they would always give me the biggest elephant to climb on, because, I know, it's a service thing of respect, but also they were always the safest. But at the end of the day, I was always super tired, hot, and basically slippery from being so sweaty. And I would get right up to this point and slip and face plant right into the elephant's butt. So as you can see, everyone's standing around taking videos, which they got and shared from every single angle, before they decided to give me a smaller elephant, thankfully. But Abby wanted serious and uplifting, so I'll try and do at least serious, maybe a little bit uplifting, and tell you about how my PhD research totally fell apart, and how we kind of turned that around and got some good data out of it at the end. So I work in Myanmar, where in the 1940s, about 10,000 elephants roamed. But after decades of catching elephants for use in the labor industry and extensive habitat loss and human-elephant conflict, that number dropped to about 2,000. So where I started work was right here north of Yangon, our primary field site, but then we also worked a couple areas across the country. My idea was to use movement data we captured using satellite collars on these elephants and monitor how elephants reacted before, during, and after conflict, and determine how effective our mitigation methods were. And then answer some broad landscape questions, such as, do male and female elephants have a different home range size? Do they use different habitat types? And most importantly, number 3, do they have different foraging behavior and conflict avoidance techniques, especially during peak human-elephant conflict times? So we started off by collaring 10 elephants in the first 2 years. We're going to be up to 28 by the end of this year, though. And we got some decent data. Each of these different color series represents a different elephant. So you can see where a lot of the elephants like to spend time, overlapping in their territories. We can see some corridors they seem to be using, and then big empty spots, why we don't know why they're going there. We were also able to show the government that when they translocate an elephant, which is basically when they just take an elephant who's causing a lot of conflict, put him onto the back of the truck, and drive him to somewhere where he hopefully won't cause a problem, such as this area right up there. But within about a week, all of the time and effort they spent doing that, they walk about 40 miles back home. But unfortunately, as you can see from this graph, the data that I really wanted was from the months highlighted in red, and I only had 2 elephants actually make it for a full year. We had a big issue, and continue to have an issue, with equipment failure, but most importantly, we discovered that elephant poaching was a much bigger issue at our field site than what we had previously thought. And so where I worked, which is that spot highlighted by the box down there, we lost a total of 19 elephants, 7 of our collared elephants, and we found another 12 uncollared elephants in that 35 square kilometer area, and over 70 elephants, including a mass grave of 20 elephants over the whole country. One of the bigger issues we found was not only are they killing the elephants, but they're not going after ivory. So they're taking the elephants for their skin and other body parts. And why that's such a big deal is because females are what drives population stability in this area. A male can mate with multiple females, but a breeding female is really important to keep the population going. So instead of doing movement analyses, I ended up doing calculations like this, where a human, if your skin was worth as much as an elephant, you'd be worth about $10,000, but they could sell an elephant skin for about $25,000. In my area, farmers make about $1,000 a year, so this is a big driver, possibly, for poaching. So, after losing almost all of my study animals, we were able to find some at least good spots at the end. We ended up publishing the discovery we made. It was one of the first quantitative studies of the elephant crisis going on in Myanmar and throughout other areas in Asia, which got also made into a educational outreach, which schools all over the country and all over the world use in their high school classes. Our collaborators at WWF used this as a rallying cry and created a whole campaign called Voices for Momo, which is an enduring term for elephants in Myanmar, that engaged over 25 million people in Myanmar alone with anti-poaching messaging. And working with the Myanmar government, we were able to help them form an anti-poaching patrols across the country. We created a bunch of community-based anti-poaching efforts, including my creative inquiry kids, who made these children's books, which we give out to about 150 rural schools in Myanmar. I was able to start up a new collaboration looking at poaching perception with San Diego Zoo Global, and they had me out there to give a talk last year, which was really cool, if you've never been to San Diego Zoo. And finally, I was able to work with Sherry Rodriguez here, and we ended up developing a novel way to quantify indirect impacts and how that contributes to human-elephant conflict, which ended up with me getting this postdoc here. But as Dr. Joda said, you can't do that without an excellent support system. So I work with all of these people, and thankfully they were there to help me out while all of the pieces were falling down, and all of our funders, which also helped. That's my story.
Slides. I'm Dr. Beth Ross. I'm an assistant unit leader in the South Carolina Cooperative Fish and Wildlife Research Unit. So my research is primarily focused on quantitative ecology. So I like to analyze data sets, interested in statistical methodology, things like that. Shockingly, I did not start off my career wanting to be a quantitative ecologist. So more of you should definitely consider that route. So I actually started off with an interest in physiology, particularly related to conservation and applied methods. So my master's degree was studying small mammal ecology in Colorado, looking at Orr's kangaroo rat and different movement patterns between linearized habitats along roadside and more natural shortgrass prairie ecosystems. So the goal of my field work, the hypothesis that I was trying to test there, was whether or not these two movement patterns differed and if they had more linear paths, and therefore if that linear movement pattern limited eventually the amount of food they could acquire in sort of an energy balance sense. So unfortunately, I didn't reject that hypothesis. So I did not detect any differences between these two surveys, or two field sites. But the other kind of underlying hypothesis I was testing at the same time was that I wanted to be a physiologist studying small mammals in an applied fieldwork setting. And I did reject that hypothesis because I did not want to do that. So at the time, I like being outdoors, I like doing outdoor things, so this fieldwork component seemed like it would be a natural fit. It seemed like I like to do that recreationally, perhaps not for an actual job. But I felt a bit like an outcast. You know, everyone around me enjoyed doing this field work, and it's great that so many of you enjoy doing field work. I'm my favorite on field work. But I wanted to kind of take this opportunity to present perhaps other options if you perhaps feel like maybe the route you're on isn't what you're looking for, whether it be in academia or wildlife ecology in general. At the time, I didn't know there were other options or avenues to pursue when I was working on my master's degree, which fortunately worked out in finding a PhD that was more quantitatively based, something that I was really interested in pursuing more, and worked out in the end. So I am happy now. To be clear, it's not like I'm... So some of the different options that I think you are being prepared for as you go through graduate school are things that prepare you well for private industry settings as well as state and federal agencies. So things like writing skills are applicable to many different outlets. whether that's grant writing, communication skills that you're also developing through speaking, and other outreach opportunities you might have access to. So those are different skill sets that you could be developing more. Additionally, there's things that you're using to analyze your data, like GIS, R, data management ideas. Those are all highly desired and sought after in private industry, as well as state and federal agencies also. So I think your advisors, I'm guessing, would be very supportive of you finding a career path that brings you happiness and fulfillment, not necessarily just something that is academically driven. Which leads me to another point that I would like to highlight, and that is that I totally disagree with the idea of following your passion, which I know is something that we often talk about in wildlife and fisheries ecology. They've actually done research on happiness in people and fulfillment, and it turns out that if you are good at a job, you actually have more happiness than if you are pursuing your passion, whatever that might be. And I'm not saying those have to be mutually exclusive. You can be really good at your passion. But thinking about how to cultivate a skill set that you are good at actually may bring you more happiness in the long term. So in preparing for this, I realized there are a couple of books that I would like you guys to have access to. So we'll be adding these to the McFadden Library, which is in Amy Sanders' office. So one of them is called Deep Work, which is by Cal Newport, so that one will be available to you. And Cal Newport also has a book called So Good They Can't Ignore You, which is about developing a skill set that can set you apart from other people. Deep Work is more about how to work in a focused manner to get good at that particular skill set. He also has a really good blog called Study Hacks that talks about these ideas as well. So that's my presentation for the day. A failed hypothesis, but an okay outcome in the end.
One of these guys, right?
It should already be open.
Down here. Yeah, there we go. Okay.
Hey everyone, I'm Sarah Bombacci. I am a new postdoc and I'm working with Dr. Jahowski. I'll be doing some work on bat and carnivore community interactions, and more broadly I'm interested in just animal community interactions, trophic cascades, stuff like that. So being new, I thought I should really make a good impression, and what better way to do that than to talk about failure. So I don't know if I got that figured out quite right, but I'm going to tell you a story about how a failure in my PhD actually turned into a publication. So
It turned into something good.
And so when I heard that I had the opportunity to talk about failure, I really jumped at the chance because, like a lot of folks, I suffer from imposter syndrome. So I've had the fortune to work with a lot of amazing people, and I still have that opportunity. But the downside to that is when you work with great people, you start to feel like everybody is perfect except you. And I only learned that that's not the case after discovering the failures of some of the people I admire. And so those experiences kind of helped me learn that failure is actually part of the process of discovery and success. And honestly, truth be told, I've learned a lot more from my failures than I ever did from my successes. So kind of to jump into what my failure story is, I need to give you a bit of background on my PhD. So I was interested in these trophic cascades and island ecosystems. So basically on islands, invasive mammals wreak havoc on the native wildlife. And so I was interested in what happens when you remove those invasive mammals and how does that affect the native island fauna. And for my PhD, I was working on birds because in New Zealand, where I did my work, they're kind of the main taxa numerically. But also I'm interested in continuing this research, hopefully throughout my career, looking at some of the cool bats they have there and reptiles. But for now, for my PhD, we're focused on the impacts of invasive mammals on birds and then the cascading effects on the ecological roles that birds play in these ecosystems. So pollination, seed dispersal, insect consumption, stuff like that. So in New Zealand, there's a lot of great data on mammals. So they're really focused there on suppressing the invasive mammals. So if you need to know if you're doing a good job at suppressing them, you need to have a lot of information on their abundance. But there's not so much data on the rest of the stuff that I was interested in. And that's okay, right? That's what I've got to go out there and do, is do field work and get that data. But the problem really came in that the models to estimate bird abundance or densities, these population models are really data hungry. They require a lot of data to get good estimates. So when you're working in a place like New Zealand, that becomes a particular problem, because in New Zealand, as this picture shows really well, a lot of the flat landscapes are gentle, brave. That's areas that have been converted to human land use, so agriculture, grazing, that type of thing. And the areas where the native forests and shrublands exist happen to occur predominantly on these really steep slopes. And so they're thickly vegetated, they're really steep, and that's where the native wildlife are, so that's where you need to go sample. And here's just another example, again, just showing that there's just these deeply incised valleys, steep ravines, and all that amounts to this terrain being really, really difficult and time-consuming to navigate, especially when you need to get a lot of data across many sampling points. And just to give you some sense for that, here's a couple of my field assistants trying to go through some of the vines and pull herself uphill using some of the vines. And so you can see that whole process ultimately is going to be really slow. And it's going to be hard to get a lot of data. So I kind of started to freak out, and this is what I thought my PhD was going to turn into. But after stressing for several days, I came up with a solution. So I knew I could get my hands on some acoustic equipment. And I've been reading about how folks have used acoustic equipment to estimate occupancy and abundance for birds. So I was like, great, I can go set some of these out at a subset of my sampling points, program them to collect data for me every day, and that saves me having to navigate to those sampling points across that terrain to get that data. So that's a great solution. But the problem is that I can't really assume that the detection process between these two different methods is going to be the same. So I can't assume that human observers and the acoustic recorders are going to detect the same birds. So I needed to come up with some way to, correct for that bias or correct for that difference. And so it turns out if you actually just carry one of these around with you when you're doing bird surveys, you really have a double observer or a paired sample where the calls and the acoustic environment, the same sounds are available to both the human observer and the acoustic recorder. And knowing that, you can measure that bias. You can measure the difference between the two and create some parameters that you can put into models to adjust for that difference. So that's essentially what I did. And I'm not going to go, for your sake, through any of this, but I'm happy to chat about it over a beer. I really do love talking stats, so if you want to know more, definitely chat with me about that. But basically, the goal I wanted you to know is there's some math behind this, and the goal is to estimate this delta parameter, and that's what you put into your models to actually correct for that difference. And so, long story short, this ended up working out great. I was able to get enough data to estimate those bird densities and save my PhD, essentially. So that's great news. But I think really more importantly, what came out of this is that I was able to get an extra publication, and not just getting that extra publication, that's great, but the reviewers, surprisingly, were very positive about my manuscript. So they really think it has a lot of potential to help increase monitoring in New Zealand. And so that was really satisfying to me to hear that from these reviewers, that they think this really can do something for bird monitoring in New Zealand, which is really important because a lot of the birds there are endangered or facing extinction. So there's a really strong need for better population monitoring. And again, it can help a lot of researchers because it's not just me that's having to go up these steep hills and collect data. Anybody interested in estimating a bird abundance is going to have to face the same challenges I did. And so hopefully, my story tells you that even if you think that you've failed it, you might be able to turn it around with, again, support from a lot of folks. I had a lot of help throughout this. It's not all just me, but with that support and a little creative thinking, you might be able to turn around and actually, you might nail it. And that's all I got.
All right. So I'm Troy Farmer. I'm fisheries ecology here at Clemson, assistant professor. And when Abby approached me today about talking, I of course had a lot of stories like everybody else, from an electrofishing boat catching on fire, to a one-year experiment during my dissertation that should have taken a year, that ended up taking 3 years, due to fish dying in the lab and all sorts of unforeseen difficulties keeping fish alive. But I thought what I talked about everyone today is my master's thesis, which started out with kind of one set of objectives and hypotheses, and about a third of the way through kind of morphed into a completely different master's thesis due to some unforeseen difficulties. And so I thought it was appropriate, given our Disasterpiece theater, to title this A Tale of Two Theses. And it really was an exciting time as a master's student to start on this project, and then at times it also felt like the worst of times when things were going poorly. But hopefully I'll end with some uplifting notes. So my master's thesis was actually looking at largemouth bass and southern flounder in a coastal area near Mobile Bay, the Mobile Tensal Delta. It's a large freshwater river delta that drains into the Gulf of Mexico. And these two species exist across this salinity gradient in Mobile Bay. And we were really interested, at the time of my master's thesis, the state was increasing their fish tissue testing program, and so they're starting to test a lot more broadly for mercury, and realizing that in these coastal areas, mercury is a big issue. And fish that, like largemouth bass and southern flounder, that people want to catch and consume. And so my master's thesis was trying to use fish otoliths to understand mercury bioaccumulation in these species. And so just really briefly, for those of you that aren't familiar with fish or otoliths, otoliths are the inner ear stones of fish. As fisheries biologists, we use them for all sorts of things. We age them as the most common use. You can see otoliths lay down these annual rings that you can really easily count and age fish. Also, a really unique property of otoliths is that they're inert, so they incorporate elements from the water and some elements from diet into the otolith. And those elements, once they're laid down, can be analyzed with laser ablation. So we put them in a small chamber, burn a transect across the otolith with a laser, and then look at the elemental composition across the otolith. We can learn something about the water that fish is inhabited across an estuarine salinity gradient that can tell us whether the fish was in freshwater or saltwater during certain parts of its life. So it's a really unique tool that fisheries biologists have over the past couple of decades really exploited to learn a lot about migration and movement of fish across different water boundaries. So the cool thing I was really excited about in my master's thesis when I started, we were going to use fish otoliths as kind of mercury indicators. There had been some neat work done by Audrey Geffen in her lab at the University of Liverpool that showed that experimentally, her students had put gobies in place, which is another flatfish, in aquariums, dosed those aquariums with set levels of mercury treatments, and shown that levels of mercury in the otolith paralleled tissue levels in the fish. So she had actually shown that you could potentially use mercury across the otolith to recreate lifetime bioaccumulation patterns in these species, which would be really neat. Instead of just taking a tissue plug and getting a mercury concentration at a set point in time, you could go back and actually look at what happens at certain time periods in life and understand how ontogenetic changes might affect mercury bioaccumulation. So that was my first thesis that I started out on. The first thing we wanted to do was go out and we were really interested in looking at environmentally relevant levels, but we wanted to find some fish that we knew had mercury in them. So we went to this EPA Superfund site that had a history of mercury pollution and got to sample there, which was an experience and a whole other story in and of itself. We thought when we pulled up fish that they might look something like this. Actually, the fish there looked pretty reasonable, but we did find that the mercury levels were about 3 to 5 times above the EPA safe consumption advisory, so we successfully collected some fish that were loaded with mercury. We then took the otoliths. I took the otoliths up to the University of Windsor, where they had a nice laser ablation ICP-MS unit with a laser here that passes a laser beam into a microscope where our otoliths are contained in this ablation chamber. It ablates the otolith, passes that ablated material in a gas form into an ICP-MS unit where you can then get some elemental composition of the earth. So it's a really neat system, and I was really excited to be using this kind of high-tech system at the University of Windsor. Like any great science, I had a really great collaborator. I was kind of coming in as a fisheries ecologist, so Dr. Joel Gagno was a biogeochemist at the University of Windsor and on my committee. And Joel was really excited about this work. I remember the first day I got up there, we had some really great meetings and talking about just how this was going to work, how we were going to. The first step was to develop a Mercury standard because there wasn't a commercially available standard available for laser ablation. We, of course, needed a standard to convert from counts per second from the mass spec into parts per million and understand how much mercury was in the otolith. So we spent the first day in the lab using a technique that Joel had used before, which is to soak some filter paper in a known solution of mercury, and then ablate it with the laser the next day. So we developed the standard this first day, and the next day we were ready with the laser. We put the standard in the laser the next morning and turned on the laser, and immediately got a nice spike that was very stable in mercury. And so we were really happy that we were going to have a nice calibration standard to convert from counts per second to parts per million. We then put our otoliths in the laser and started to ablate them and realized that our mercury was not moving at all. It was consistently high. And so then we tried to flush the mass spec with lots of argon carrier gas for several minutes, which turned into 30 minutes, which turned into an hour of just trying to flush the system. And over a series of time, after replacing all the tubing, after cleaning the mass spec, we essentially realized that we had contaminated the entire mass spec with ridiculously high levels of mercury. So I had flown all the way up from Alabama to Windsor, had a hotel booked all week, and was going to collect a lot of my data for my thesis. And on day 2, we had basically ruined the mass spec. And so needless to say, those guys in the graduate students in the lab were thrilled that the mass spec was down. Had technicians come in and do all sorts of cleaning and recalibration to get our mercury out of the system. And so I went back to Auburn University with no data and really nothing to show for this big trip and this big expenses I incurred to fly up to Canada and process samples. So at that point, we said this thesis is not going to be feasible. We need to figure out something else to do, like everybody else. I had some great collaborators and a really nice committee. And we kind of revisited the question, what can we use fish otoliths for? And fortunately, in reading the literature and being familiar with sort of the many uses, there were some nice common elements that we could use as habitat indicators in mercury. Strontium and barium, as I mentioned again, to look at where the fish had resided in its life. We had tissue plugs from a lot of these fish I had sampled in the first year of the project. So we had mercury levels, and we were able to kind of establish the project by using strontium levels in the otolith as an indicator of salinity exposure. And this turned out to be really cool. It resulted in a couple of publications that are still getting cited and some neat hypotheses we developed. The thing we found out about residents in the Mobile Tensile Delta is that if you were in the freshwater portion of the delta, fish had a lot higher mercury than in the saltwater portions of the delta, and the brackish water portions. And what you can see here is for largemouth bass, which are shown here. I think this is the H1, H2, and H3 fish. There's strontium to calcium in the otolith, which increases as salinity increases, was negatively related to their total mercury body burdens. So we saw that fish that were in the freshwater portions of the delta had a lot higher mercury, and those that spent a lot of time in the brackish water had a lot lower mercury. I was able to go out and collect some diet samples and explain why this was with different trophic dynamics and different potential for methylation in those two areas. And it resulted in kind of a better understanding overall of how habitat-specific resonance patterns influence fish tissue contamination. So, and it also led to a much greater understanding of flounder migratory patterns across salinity gradients. We kind of came into this thinking there was this accepted life history pattern where juvenile flounders move into the estuary and move towards freshwater very early in life. And then as they grow and get larger, they slowly move back down towards saltwater. We found out that some of these flounder actually go up into the freshwater portions and stay there virtually their entire life until they're 2 or 3 years old, and they all of a sudden look like they run very quickly back out to salinity to spawn later in life. And that led to a project here at Clemson with Jared Crisp and Megan Angelina, following up on this work as master's students to go out and kind of try to understand how widespread these kind of divergent life history patterns are for southern flounders. So even though we destroyed a mass spec, we still got usable data, and we're able to provide some interesting information. And a couple years ago, some people that were currently smarter than us figured out how to detect mercury in the otoliths, so it is possible, but not using a laser. So with that, that's my story of kind of perseverance and overcoming some odds. So, thanks.
All right, well, that actually concludes our first ever Disasterpiece Theater. So I think the plan is to go to Nick's after this. So hopefully we'll see you guys there. Thanks.